Hi, we will talk about the collective behavior of cellular actors of inflammation. And to do that, first of all, we will try to understand some basic principles and mechanisms uh, in the process of the inflammation, because without that part, it will not be possible for us to model the process. Simply said, inflammation is a generic response of the immune system to an event of injury or infection in an organism. It is manifested by five so-called cardinal signs of inflammation. I will not dive into it. But what I would like to say here is that there are two types or two stages of inflammation. The first one, the acute one, is sudden and lasts just for a few days. And in case the immune system does not succeed to resolve the issue, then we will move to the chronic stage of the inflammation, which lasts generally many months or even years. It is important to study inflammation because it will help us to understand how it happens and to find solutions to reduce the burden of numerous diseases that target many individuals among us. The mechanisms of inflammation start with the event of injury or infection, which is materialized in the tissue by the presence of the damage associated molecular patterns in case of injury or pathogen associated molecular patterns in the case of infection, which constitute, I mean, those two molecules constitute what we call here the primary chemoattractant. And a chemoattractant is simply a small molecule that bind to the receptors of leukocytes and cause them to be stimulated and to move um, to move or migrate towards a certain or a given direction. The presence of these primary chemoattractants will activate the resident immune cells or scouting new immune cells present in the tissue via their pattern recognition receptors. This activation, once activated, the immune cells will secrete and release in the tissue inflammatory mediators, among which we have the secondary chemoattractants. We will only focus on the secondary chemoattractants here because this is the one that is important on the process that we are modeling. These chemoattractants, secondary chemoattractants, will be the origin of a series or cascade of recruitment right from the blood vessel of immune cells, typically neutrophils, because they are the first responders, and their migration to the site of injury. They will migrate there to solve the incidents that occurred in the organism. And this resolution consists on the removal of intruders or debris, generally through phagocytosis or maybe through other mechanisms, and also the degradation of the inflammatory mediators in the tissue. Once you have discussed and understood these basic mechanisms of the inflammation, we can now move to the modeling part where we identify two main components, the chemoattractants and the immune cells. The chemoattractants are synthesized, as we just said previously, by the neutrophils and they diffuse and decay in the extracellular environment. And the immune cells will move towards the increasing concentration of chemoattractants or towards the gradient of chemoattractants, but this motion will be also subjected to small deviation due to other functions of the immune cells or also due to the uneven structure of the tissue. And this deviation can be modeled by adding a random component to the trajectory. The change of concentration of the chemoattractant can be assumed to have two origins. The first one is the diffusion. in the tissue. And the second one is the which is represented here by the function F, is the change due to the production and the decay of chemoattractants in the tissue. This production take into consideration those MIs, which are the intracellular quantities which influence the amount of chemoattractant synthesized, because in reality, in biology, there is a lot of complexity inside the cell. There is a cascade of reactions that are happening, and you can imagine that there are some cells, some molecules that may influence the production of attenuate or inhibit the production of the chemoattractant. And since we consider the intervention of these intracellular quantities, it is also important to model their change by an arbitrary function that I have represented here as Ji. So, coming back to this function F, it is here. It is represented here with this gamma. This delta P X, which accounts the fact that we are computing or we are calculating this production at a neighborhood P of a given cell X. And yes, we are summing up all these productions and decay with the lambda rate to all the cell at all the cells in the tissue. Sorry. The production term here take into consideration the concentration C of chemoattractant sent by the cell, but also another parameter C1, which represents the sensitivity of the receptors of the immune cells to the gradient. And we can easily agree that for large value of n, C1 represents here the threshold for the production of the chemoattractant. The second factor here, G, represents simply the attenuation factor of the production of the chemoattractant. This series of equations can be solved using numerical methods like finite volume methods or anyone else that you can find in the literature, but solving these methods is not in the scope of this lecture. Going back to the second component, which is the cells, the immune cells and their trajectory. The change of their trajectory has here three components as the time is going on. The first component is the contribution of the chemotactic force towards the direction of the gradient. But I would like to emphasize here that grad here, the appellation grad does not mean the amplitude of the gradient, but simply the amplitude of this chemotactic, of the contribution of this chemotactic force towards the gradient. The second component, which is called here ramp, just add the noise or the randomness as we discussed earlier. The last component here, which may be the less obvious, simply take into consideration the fact that we assume that the cells are not stupid enough to move from one step to the other step in two completely uncorrelated directions because of the randomness that we have assumed previously. Yes, cells are smarter than that. So this assumption or this persistence of the motion is taken into consideration here by this coefficient. But yes, all these values can be fine-tuned in the model to better represent the observation. And when we have this model, we can now go to the simulation, where we have this simulation here of a very specific case where there is no intracellular attenuation factor. You can see with me that the NIs have disappeared. And here, if we simulate it in a three-dimensional environment, where the green spots here represent the neutrophils, and we have that red spot which represent the chemoattractants at the onset of the inflammation. We can observe how it spread, how the chemoattractant spread in the tissue, and how the immune cells migrate toward the site of infection. And interestingly, we can observe here, for instance, in these two figures, how the immune cells start to produce in the tissue some chemoattractant, and that help the spreading of the signal. That's all for today. Thank you.